Good afternoon. My name is Joe MULDOON. I'm with a company called FASTBioMedical, and Peter is a tough act to follow, I got to tell you. But my goal in life now is to change his last slide from what is your serum creatinine to what is your measured glomerular filtration rate. A little bit of a preview of what we're going to talk about with FAST. But before I get started, two things. Thank you to the leadership of AAKP for inviting me. It's an honor to be here. It's always a thrill to talk with you. I don't know how many of you had the misfortune of hearing me speak at the annual meeting, but here's what happened. Got to slide 3, it froze there, and I'm blabbering on. I'm on slide 4, I'm on slide 12, and people's eyes are glazing back in the audience because I didn't know it froze. So I'm going to pay better attention to that, but if I'm still talking and the slide resembles nothing what I'm talking about, like give me a sign, like throw a napkin or something. And then the second thing, I have a little bit of a reputation of maybe being candid to a fault sometimes. And I noticed that AAKP asked me for the slides and I sent them in, and then they said, oh, by the way, we're going to be taping this. So I'm sweating a little bit. We're working on a first-in-class technology that doesn't estimate your kidney function. It actually measures your kidney function, and we measure your volume as well. Currently, there is not an approved product that is clinically viable to do that. The FDA has determined that we meet the requirements for an expedited review. That's pretty rare. And to date, we've received substantial NIH support for which we are eternally grateful. We think this is going to give us the ability to move past trailing indicators and pretty crude surrogates of both kidney function and volume status. Today, we have estimates of kidney function. eGFR is the standard of care based on serum creatinine, and I'm going to show you what the limitations of that are in a minute. And then we have estimates of fluid responsiveness, but we don't have a direct measurement of volume that's clinically viable. It's important to point out that this is an investigational product at this point, not yet approved for human use. Serum creatinine, poor marker of function. Let me give you one example in AKI. So in this example of an AKI patient, we're tracking their serum creatinine. And it kind of has a nice gradual increase over several days. It's because it's such an insensitive trailing indicator. Matter of fact, you can lose well over 50% of your kidney function before your serum creatinine even bumps outside of a normal range. So when we're using that to determine our estimates of GFR, in this scenario, we also have a nice gradual slope reduction in kidney function over days. Well, when you take that same patient and you compare it to their measured GFR, you find that what happened is the kidneys had an insult and the GFR, the kidney function, dropped off the face of the earth. And not only did we not know that, but probably a lot of things that we were doing to this patient, including dosing them with nephrotoxic antibiotics, was kicking the kidneys while it's down. So we think measured GFR can be an important part of moving this field forward. When we look at some of the calculations of eGFR and the move from MDRD to CKD-EPI and what an improvement it was, I think it's important to point out the data that was used, even in this publication, which was a great publication. The gray dots are individual measured glomerular filtration rates. They used a clinical research standard believed to be accurate, I believe it is, to measure those GFRs. And then they compared that to the population-based estimate of eGFR. So my observations are that's a really extremely wide spread from the mean. eGFRs can be plus or minus 30% of what your actual GFR is. That's as accurate as we've been able to get this estimate. So you can get an estimated GFR of 50. Well, what does that mean? Well, it means it might actually be 35, of which you have severe disease, or it might actually be 65, of which you're in stage 2 CKD. That's a big difference. It's a big difference that we don't know in real time clinically today. I believe that eGFR is useful and it's accurate across patient populations, but I would submit to you that it can be very inaccurate on an individualized patient basis. So if we're really going to move towards precision medicine in kidney disease, we need a way to measure GFR. We have that. The way it works is it's a combination of an injectable and a device. The injectable's proprietary, we developed it. The device is proprietary, we developed it. We give the patient a dose of the injectable. We call it VFI. We give them a very small amount of it. It's 3 milliliters in solution. In the 3 clinical trials that we've done, we're putting it in an IV in their arm. It's a single bolus injection. It's one single push. It's predominantly dextran. Dextran's got a really strong safety profile. It's been used for 50 years, and we're giving patients a very small amount of it that's fluorescently labeled. What that does is gives us the ability to see their renal clearance by one component of the marker and establish their volume by another component of the marker. So if you want to know the volume of that patient, you need one blood draw at 15 minutes, and we can calculate their plasma volume. If you have plasma volume and you have hematocrit, which is normally taken, you can also calculate their blood volume. If you want their measured glomerular filtration rate, you take 2 more blood draws over 170 minutes, and we can calculate the GFR. What clinicians have loved on the plasma volume measurement is that once you dose that patient, you can calculate and recalculate their volume for 6 hours. So they can see where a patient is, intervene with fluid, and then if they're wondering if it worked, just take another blood draw and it will give them an updated volume. So it remains resident in the vasculature for 6 hours. You don't need to redose the patient. For kidney function, you do need to redose the patient. We have a validated fluorescence assay and a validated HPLC reference assay, which we've used in our clinical trials. Our vision of the commercial form is a direct fluorescence assay that could be used in the lab or pulled closer to the bedside of the patient. We think there also exists the potential for it to work on existing lab instrumentation, but we've not developed that pathway yet. So how might that impact patients? Well, in transplant patients, we think it has the great ability to give you a tool to better evaluate living donors and see precisely what their function is. As we've talked to transplant physicians and CEO of transplant facilities, they were particularly excited about in cadaver donors, where some of these people are young, it's a result of trauma, but they still have them on a closed circulatory system, so our technology would work. And they said in a lot of these patients, we have literally no history of what their renal function's been, and in yours, in real time, we can see how these kidneys are working. They were also excited about the volume status because they said keeping that cadaver at a normal volume level while we're making these assessments, we believe will optimize our ability to actually harvest as many organs from that patient as possible. Peter talked about, post-transplant surveillance, which is obviously very important. We think it gives us another unique tool. So in addition to a marker of injury, now you'll have a marker of function. You'll be able to see in real time how is that transplanted kidney functioning. And you'll be able to optimize the dose of immunosuppressants. I've always found immunosuppressants ironic, right? If we don't give the patient enough of them, the body kills the kidney. If we give them too much, the drug kills the kidney. They're renally cleared, then they're nephrotoxic. So the one thing you'd want to know is what's the impact on kidney function and what's my renal clearance, and we give both of those. Other benefits really for all renal patients, better dosing of renally cleared drugs. So the majority of drugs are renally cleared, and because we don't have a way to accurately measure renal clearance, by definition, we're overdosing or underdosing probably 2/3 of those patients. It enables the ability to measure renal reserve, which is believed by many of the key opinion leaders in the space to identify risk of progression, is knowing what a patient's renal reserve is, which we don't have the ability to measure today. We think it could be a better endpoint for clinical trials, so move us past death and dialysis as endpoints and actually say, what did it do to their measured glomerular filtration rate and over what period of time? We think it could potentially detect early onset of renal dysfunction, so you can better stage patients for treatment and trials. As you probably know, EGFR over 60 has no meaning. They don't track patients over 60. Wouldn't it be nice to see a patient that's above that level, but that is starting to lose function, so we can intervene more early? Dialysis patients, we think the volume status can help better dial in their dry weight, which is believed to be the reason why dialysis patients feel so awful after dialysis, is because we've made them hemodynamically unstable, but we don't have a way to measure what stable is or how far they are off from it today. More importantly, we think understanding measurement of function accurately in real time can help us delay or avoid even the progression to ESRD. AKI patients, particularly useful because serum creatinine is not useful at all in that market because it just can't pick up dynamic change where this can, and give you the ability to measure response to therapy. And then last but not least, in cardiorenal patients, we're trying to understand the impact on the cardiac system, where volume has a huge role, and the renal system, where obviously you have MGFR. We think it's a really unique tool in that patient population. So our clinical history, we have conducted 3 trials to date. We've dosed 89 patients. We've given them a combination of 97 doses. We've conducted our trials in Germany and different sites in the United States. We've tested in healthy volunteers and through stage 3 and stage 4 chronic kidney disease. Lots of comorbidities were involved, including hypertension, diabetes, and COPD. We proved that our injectable is very well tolerated. We didn't have any SAEs. We also demonstrated extreme accuracy, repeatability, and sensitivity, and we achieved both our primary and secondary endpoints in those trials. The most recent trial data on the kidney function side we now have published in JASN, and I'll give you a glimpse of that in a minute. And we've got a manuscript in process regarding the plasma volume data. This is an excerpt from the JASN publication. We dosed 32 patients, healthies in stage 3 and 4 CKD, and what we did is we compared it to iohexol clearance. So iohexol clearance, as you probably know, is a research method that accurately measures kidney function. It's not used clinically because it takes too long, especially to get the results. It takes a couple weeks usually, so it's just not quick enough to make a therapeutic decision on. We had a 0.9961 correlation to iohexol clearance across all renal function levels, so we were pretty excited about that. For us, next steps on the regulatory approval and clinical trial plan, we're looking right now at a potential trial in Germany that we've been invited to that would get us in 50 heart failure patients, which is also a patient population that could benefit from this tool. We believe we're one pivotal trial away from approval. Capital dependent, we'd like that to be a multi-site, multi-country trial, because our experience has been in Western Europe and in the US, and that they're asking for pretty much the same things. It'll be 36 months from closing of our next round of financing till when we think we have this product in the market. Kind of the long poles in the tent are scaling up the injectable, building it in bigger and bigger batches and putting that on stability. That's about a year-long process. We're preparing for our pivotal to be about a year, and then we'd file our PMA for approval. So now when I look past kind of our technology and more broadly into the market and what do we see happening in renal, and try and kind of relate it to what we heard from others, entrepreneurs, Sandeep, as he's talking about the Kidney X Fund. I go back to this article that was put out in JASN by some folks at the Kidney Health Initiative addressing barriers in kidney health. And I just took out one excerpt. Kidney disease is a global healthcare epidemic. However, we have fewer randomized controlled trials than any internal medicine specialty. and we've received less funding from public and private sources compared with other diseases. I'm a data guy. A couple of the slides that Sandeep showed you were slides that I pulled from this deck that I sent to him. So when we look at, so where is private equity investing happening in healthcare? I look to, this is one report, it's from Silicon Valley Bank. They produce it twice a year. So they just produced the first half of 2018, where they look at venture investing in healthcare. Well, I can tell you there's plenty of it. 2017, $9 billion, record year. 2018 looks like it's on the same path, $4.5 billion for the first half of the year. So there's plenty of money going into healthcare. Unfortunately, it's not coming into renal. If you look at the top areas that get funding on the drug side, oncology's number one, but renal's not anywhere on the list. It's not on the list of the top 25. Orphan and rare disease, which by its definition is rare, gets more funding, more equity funding than renal disease does. When you look to device investments, it's worse. Dermatology gets more funding for innovations than renal disease does. Why? Because investors like to make their money back with a reasonable return for the risks they took, and the exits do not exist in renal. If you look at device exits on the top, cardiovascular leads the circus. If you look at drug exits on the bottom, oncology leads the track. Why? I've got a bias in this, but I've heard oncology and cardiology come up at least 4 times today as examples of where innovation has been ushered in. If you study those fields and see what the tipping point was, the diagnostics got better first. And when the diagnostics got better, then you saw a wave of therapeutics and interventions come in because we actually knew which patients to do it to, and we could actually see whether it was working or not in real time. So I think we need to double down on the diagnostics in this space. A summary of what we covered today. We believe we're on the edge of moving from really crude estimates and trailing indicators of kidney function and volume to the real thing. We believe it can favorably impact transplant patients and the broader renal and even cardiovascular community. Our technologies are in late clinical stage development, and the renal area and kidney disease is grossly underfunded compared to other disease areas. And that's one of the reasons, that's in my mind, that's the main reason that innovation has been stifled in this area. That's all I got.